people, the other stragglers coming in. I'm Nelson Lau, Director of the Genome Science Institute, and I'd like to welcome you all to the first GeoSci seminar of the new year, 2026. So happy New Year to you all. And it's my great pleasure to introduce Dr. Craig Ceol from UMass Medical School, or UMass Chan Medical School now, from the Department of Molecular Cell and Cancer Biology at UMass Chan. Craig is an associate professor there, where his lab uses zebrafish as a model to study melanoma, although I've heard he's also switched over to cell culture now. I've known Craig and his work when he was a postdoc in Len Zon's lab, really developing this BRAF overexpression model in zebrafish. And that work enabled Craig to get a K99R00 award that he used to then bring his lab over, to start his lab over at UMass in 2010, is that right? Yeah. He's won some Junior Investigator Awards for starting his lab, the Kimmel Scholar Award and the Department of Defense Cancer Career Award, as well as the American Cancer Society Research Scholar Award. But more recently, in 2020, you were awarded a Rising Star Award from the International Federation of Pigment Cell Societies. I didn't know there was a Pigment Cell Society.
Oh yeah.
So that's
Devin, I can tell you all about it.
Oh, okay. I've actually known Craig a lot longer than that, more than 20 years ago, when we were both grad students at MIT. I was a grad student in David Bartel's lab. Craig was a grad student in Bob Horvitz's lab. And he was an expert in staging C. elegans worms, and I was a total novice. And I was trying to get C. elegans to see if we could discover more small temporal RNAs like LIN4 and LIN7. And Craig graciously, dropped everything and helped this newbie grad student collect worms grown in liquid culture, and I will forever be thankful for your help in that. And so we had these, formative years as grad students at MIT. But then we kind of diverged paths for quite a bit. But I've invited Craig back to Boston because I've become interested in fish skin biology. And so I'm hoping to hear some of that, as well as maybe strike up a new collaboration there. So welcome, Craig. Looking forward to your talk.
Thanks. Is this volume is good? All right. So I'm really happy to be here to give this talk, and part of it is because I have a lot of respect for the science that's being done here, and part of it is because I get to catch up with people who I've known, over the course of however long I've been doing science. Neil Gainham's lab and my lab, we've had collaborations. So in the recent past, I've known Deb for a long time. Through Pigment Cell Society and Hui from Zebrafish Society. And like Nelson said, you didn't quite steal my whole story, but we've known each other since we were grad students. And the way that I remember it is that, I'm in lab and this guy shows up and he's like, oh, I kind of want to get some RNAs, we're just interested in looking at these RNAs. And if I remember correctly, at the time they were called tiny RNAs.
Yep.
Tiny RNAs. And so then, I helped Nelson out. I had done RNA isolation before, but I do remember you were adamant. You were like, I want all the RNAs, no size selection. And so then, Nelson goes off, and what was amazing is just a few months later, out comes this incredibly groundbreaking paper where you guys had discovered microRNAs. So that was really cool. So we've known each other for a long time. Anyway, so let me get into the talk here, and feel free to ask questions in the middle of the talk. Doesn't bother me at all. What I'm going to tell you today is the work that my lab has been doing looking at bone morphogenetic protein signaling in melanoma, and our efforts to try to target that translationally. So to get you acclimated to melanoma. Melanoma is a disease that arises from melanocytes. These are the pigment-producing cells in our skin. They normally reside in the basal layer of the epidermis. They make the pigment melanin organelles called melanosomes, which then get transferred to keratinocytes in the skin and give each of us our own distinct pigmentation. These cells can divide and form benign lesions, clinically known as nevi, but colloquially known as moles. And then these cells, they can transform and form tumors. Some of them come from nevi, some of these tumors come from nevi, but probably the majority arise de novo from single melanocytes or melanocyte stem cells. So the tumors, they start growing radially within the epidermal compartment, and they can transition to a vertical growth phase where they go and invade through the basement membrane into the underlying dermis, and then finally they can become metastatic. And I think you all know that melanoma is the deadliest skin cancer. It's by far the deadliest skin cancer. And a big reason for that is because very early on it can become aggressive. And this kind of highlights how the extent of that. So melanomas are staged in part based on the depth in which they go into the skin. And there are some that are very shallow. Patients that are diagnosed with a melanoma that's very shallow, they have a very good survival rate. But if that melanoma grows, let's say to 4 millimeters, you can see that the change in the 5-year survival rate is pretty dramatic. And what that means is that this small bit of growth in the depth says that essentially at this point, there are many tumors that have already started to metastasize. And you don't get them, you don't get rid of them through surgical excision alone. In addition to that, melanomas are staged based on their presence in lymph nodes and distant metastases. So the treatment of melanoma has really been revolutionized with the advent of immunotherapies, immune checkpoint blockade therapies. And these therapies were trialed in melanoma. And just this past year, the sort of a landmark 10-year study was published called one of the keynote studies. And I'll go through a little bit about the results from that study just to get you a sense of what the good things are about immunotherapies and what else needs to be done. So what you're looking at here is a progression-free survival curve. And in this study, what was done was a comparison of patients on ipilimumab alone. This is an anti-CTLA-4 monoclonal antibody versus patients that are on nivolumab. This is an anti-PD-1 antibody, and patients that are on the combination of both. So IPI really doesn't have much of an effect. If you don't treat patients at all, or if you treat them with sort of legacy chemotherapies, you see something like this, where stage 3 and 4 patients, only about 5% of them are still alive, progression-free after 5 years. But nivolumab has a pretty significant effect, and the combination has an even better effect. So at the end of 10 years, you have about 30% of patients that are still alive, as compared to 5 or fewer percent on older therapies. And so this is a huge, huge step forward in the treatment of melanoma. And so what you can see from this curve also is that patients basically, when you see these curves drop down, that means that patients' tumors are progressing. But if patients make it to about 3 years without progression, their prognosis is actually quite good. So you see these patients at 3 years. If they're not progressing, then they're pretty much guaranteed to make it to 10 years. And it's likely that many of these patients, even though they had stage 3, stage 4 disease that had already spread, it's likely that many of them are effectively cured of their disease. So that's a huge step forward in terms of treatment. The therapies have also been trialed in earlier stage disease. So this is stage 2 melanoma, where the melanomas have invaded. There's still a primary tumor in the skin, but they've invaded a little bit further. And you can see this is a trial with pembrolizumab. This is another anti-PD-1 monoclonal antibody like nivolumab. And there's a gap here. So that means that patients that are receiving pembrolizumab have some benefit as compared to the patients who are receiving placebo alone. But there's a big issue, there's a few issues with this type of treatment. So essentially what this shows is that yes, some patients are receiving a benefit, but it's only about 5 to 10% of those patients are receiving a benefit. And so the question clinically is, do you treat every patient, even though only 5 to 10% would benefit? These drugs are very expensive, that's a problem. The other problem is with each of these immune checkpoint blockade therapies, and especially with combinations, there is a high risk for immune-related adverse events. And so in this case, let's say that you decide to treat everybody, it's going to be expensive, but let's say you do that, you're putting patients that might otherwise be fine at risk for these immune-related events. And these things are like colitis and other types of autoimmune type of events. And some of them can be life-threatening. So there's a lot of work that's being done in the field to try to identify the subset of stage 2 patients that would benefit from ICD therapies. We can also look at melanoma trends over time and what the unmet need is. So what I'm showing you here is the rate of new cases in melanoma as compared to the death rate. The death rate is somewhat similar over time. There's a slight dip here, and that's because of the advent of immunotherapies. But what's been happening with melanoma is that the incidence has been rising steadily over the last 30 years or more. And so what that means, this is the death rate again. You can see that there's a dip here. This is due to the use of immunotherapies. What's been happening over the last few years is that there's a plateau that's being reached. And so there's a lot of patients that aren't benefiting from immune checkpoint inhibitors, and their tumors are resistant to these drugs. And you can also see some unmet need if you look at total deaths. So the total deaths is rising in conjunction with the rate of new cases. Here's where immunotherapies came in. But again, because you have this increase in the rate of new cases, the number of total deaths is increasing as well. And it's just going to keep going up. And so there is some unmet need. And a big question out there is, how can these immune checkpoint blockade resistant tumors be targeted? And this is something that we've been interested in, a lot of people have been interested in. And it sort of overlaps with the work that we've been doing in the lab, which is to try to identify new genes, new proteins that are involved in melanoma initiation and melanoma progression. And so with melanoma, recurrent melanoma, other cancers, especially solid tumors, recurrent mutations point to genes that are either oncogenes or tumor suppressor genes. And in melanoma, about half of all tumors have mutations that overactivate the BRAF serine threonine kinase. There are 15 to 25% of tumors have NRAS mutations, and another 10 to 20% have NF1 mutations. These are mutually exclusive. A tumor tends to have only one of these mutations, but they all affect RAS-MAP kinase signaling. They all overactivate it. And pretty much just about any melanoma out there is going to have a mutation that ramps up RAS-MAP kinase signaling. This is what you find in nevi, so these benign lesions. About 80% of them have BRAF V600E mutations. These are these activating mutations. But that says that these mutations alone aren't sufficient to give you a melanoma. You need other things that need to happen. And so we've been interested in what some of those other things are. And so for probably about a dozen years ago or so, we were looking at genes that were subject to copy number variation in the melanoma genome. So in solid tumors, many solid tumors are aneuploid, and if you look at copy number across the genome, what you find is that there are regions of the genome that are either recurrently amplified or recurrently deleted. So this is an example of a chromosome that's recurrently amplified in red. What you have here is the right arm of this chromosome is recurrently deleted, and so on. And so in these regions of amplification or deletion, there lurk oncogenes or tumor suppressor genes. And the trick is to try to find amongst the many genes in those intervals that are either amplified or deleted, how do you find which genes are the important ones? And so there's been work done looking and identifying the CDKN2A locus that encodes the P16 and P19ARF tumor suppressors. And this is recurrently deleted in many melanomas. You see the left arm of this chromosome here. And it's not quite as evident, but there are melanomas that have focal amplifications of cyclin D1. So these are some of the things that have been identified. But you can see that there's many regions of the genome that are recurrently amplified or deleted that we really don't know a whole lot about. And in order to tackle this problem, what we did was to use a zebrafish model, and this is a model that combines this very common oncogene, BRAF V600E, with a P53 mutation. So wild-type animals, look here. Here you can obviously see where the tumors are, and pretty much every fish that has this genotype is going to develop a tumor. But although they have a BRAF oncogene, they have a P53 tumor suppressor mutation, it's not as though the fish develop tumors everywhere, where every melanocyte or every melanocyte stem cell gives rise to a tumor. In fact, they develop tumors that are focal, so only a few of them are actually transforming. And the reason for that is because there are other things that need to happen in order to make a tumor. And those other things are copy number changes. And so we showed this a while ago. And so we were interested then in looking at copy number changes that were in these zebrafish melanomas and comparing them to what you find in human tumors. And so we did that, where we identified the genes that were recurrently amplified in human melanomas and overlapped them with the genes that were recurrently amplified in zebrafish melanomas. And so what we found is that the number of genes in this area of overlap were much higher than what you would expect by chance. And what that says is that a lot of the same genes are being amplified in both species. And that also says that the same mechanisms that are at work in human melanomas, at least some of those are also at work in zebrafish melanomas. So we thought that this region of overlap was a good place to look for new oncogenes that we could study. And I'm going to cut to the chase. This is work that was published quite some time ago. We identified a gene called GDF6. It's also known as BMP13. This is a BMP ligand. And there are many reasons why we focused on it. One of those reasons is that this gene is highly expressed in melanomas, but not in normal melanocytes. So it's tumor-specific expression. Like I said, it's recurrently amplified in about 40 to 50% of melanomas. And so those are some of the reasons why we got interested in it. And so GDF6 is a BMP ligand. And I'll just sort of take you through the BMP signaling pathway here very briefly. So BMP ligands, like GDF6, what they do is they bind BMP receptors. This causes these receptors to heterotetramerize. Type 1 receptors get phosphorylated, and they in turn phosphorylate SMAD 1, 5, and 8 proteins. And so when you see phosphorylated SMAD 1, 5, and 8, which I'll show you a little bit through this talk, when you see that, you know that the pathway is on. This is sort of the canonical marker of BMP signaling. Phosphosmad 1, 5, and 8, they bind to SMAD4, they go into the nucleus, and they regulate transcription of target genes. And they can either upregulate or downregulate depending on what proteins they're interacting with. So we did a lot of functional studies to validate that this is an oncogene. We showed that upregulation of GDF6, it promotes growth of tumor cells in culture, it promotes growth of tumors in vivo. The knockdown has the opposite effect, both in vitro and in vivo. We showed that it acts through the BMP pathway by showing that using genetic epistasis experiments to show that it acts through SMAD1. And then we also showed that what it's doing in part is to promote melanoma cell survival. And the way that it does that is it represses the cell death promoting gene SOX9. And so that's what it's normally doing. It's upregulated, it's repressing SOX9. When you get rid of GDF6 and BMP signaling, SOX9 goes up, and that ultimately kills melanoma cells. But we were also interested in another feature of what GDF6 and what BMP signaling does in tumors. And so we did transcriptomic studies to identify genes that are regulated by this pathway. And I'm not going to go through the design of the experiment, it'll take too long. But basically the pathways that we got out, these are the pathways here, and it's bone morphogenetic protein signaling, so finding ossification, that's not very surprising. But a close second was neural crest. And so this is very interesting because the neural crest is the embryonic tissue from which melanocytes are derived. And so this was interesting to us. And the features of these genes, these are neural crest genes right here. The features of them were that in control melanoma cells, they're on. When we knock down GDF6, they go down. When we have knockdown but then restore BMP pathway activity, they go back up. And so those are the genes that we were interested in, lots of neural crest genes in there. So the idea was that what they're doing is, what BMP signaling is doing is it's promoting a neural crest identity in melanoma cells. And when you withdraw GDF6, you lose that neural crest identity, and the hypothesis was that the cells would then start to differentiate. And so we looked at that, and what we found is that that was indeed the case. So MITF, this is the master regulator of the melanocyte fate, and when MITF levels are high, that will cause melanocyte progenitors to differentiate. So this is how it normally works. In the case of tumors, when we get rid of BMP signaling, MITF levels go up. So this is an shRNA of GDF6. Levels go up. MITF regulates a whole suite of genes that are involved in differentiation, the genes that go on to make melanin and give cells pigmentation. And one of these genes is TRP1. So when we knock down GDF6, MITF goes up and its targets also go up. So this is basically when you get rid of BMP signaling, these cells start to differentiate. And what I told you earlier is that ultimately they die. So that's what this is doing. We also looked at the loss of GDF6 in our zebrafish model. And so this is a case where we have our BRAF B53 fish, and they're getting tumors here. So this curve is going down as they get tumors. When we add in the loss of function GDF6 mutant, what you find is that not as many tumors form. So it's affecting tumor initiation. In addition, the tumors that come out, they're highly pigmented. So again, when you lose BMP signaling, you get differentiation. You can see it very clearly here. So they're very differentiated, and they also don't invade quite as much. And when we look at these differentiation genes in these tumors, like MITF, you could see in the GDF6 mutant tumors, they're higher. TRP1 is also higher as well. So again, you lose BMP signaling, and the cells start to differentiate. And this is sort of the overall model that we formulated to explain how we think GDF6-driven BMP signaling is working in melanoma. So you have the GDF6 locus that gets recurrently amplified, and this causes, by means that we don't exactly understand, but it causes GDF6 to be produced. That gets secreted by melanoma cells, and then it acts on those very same melanoma cells to stimulate pathway activity. SMAD1, 5, and 8 gets activated, it inhibits cell death and inhibits differentiation. So that's sort of the historical overview, just to get you up to speed on where we were at this point. So we were also interested in what the normal function of GDF6 and BMP signaling was in melanocytes. And one of the things that piqued our interest was that we had these GDF6 mutants, and it was very obvious that these animals are a lot darker than normal zebrafish. And the reason that they're darker is that they have too many melanocytes. So they have an excess of melanocytes. So basically there's something going on. GDF6 has a normal function in regulating melanocytes somehow. So we started to look into this, and we found that GDF6 embryos, they also have an excess of melanocytes, and that GDF6 expression, it was known that GDF6 is expressed very specifically in the neural crest, but only during the time when the neural crest was there and when melanocytes were being developed. So again, melanocytes start out as neural crest cells, and then they then differentiate and go through a process of melanocyte specification and differentiation. The neural crest makes other cell types, and so it's making not only melanocytes, but lots of other cell types as well. So we did experiments where we used a BMP inhibitor, a small molecule inhibitor called DMH1, and we treated animals only during neural crest, when the neural crest was present, and we found that we could phenocopy the effects of a GDF6 mutant. So you could see here that there are excess melanocytes when we treat with the BMP inhibitor. So that says that this is the time period that's really important for the phenotype that we're seeing. And the way that we teased this apart was to do some FACS experiments where we had cells that were double labeled with crestin and with MITF. So crestin marks all neural crest cells, and MITF MITF marks the cells that are going to become melanocytes. And so the experiment here was to look in control animals versus BMP treated animals and see, as we go along here, what proportion of the crestin cells are turning into melanocytes. And in control cells, you could see here that the double positive cells, about 7.8%. In the BMP treated cells, it's about 15%. So what this says is that you have this pool of neural crest cells, and when you lose BMP signaling, a larger portion of those cells are turning into melanocytes, okay? So what BMP signaling is doing is it's preventing cells from turning into melanocytes. It's preventing neural crest cells from turning into melanocytes. And the way that it does that is, as we showed in melanomas, when you lose BMP signaling, MITF levels go up. So this is a similar experiment to what I showed you in melanoma cells, but here we're looking at zebrafish neural crest cells. They're marked with crestin. When we have the BMP inhibitor present, you could see that the levels of MITF are higher, and what that means is that more cells turn into melanocytes. And the reason why I was talking about this is that the effect of BMP signaling, what we see in tumors, is that it's preventing differentiation. But this isn't some sort of effect that's been cut out of whole cloth. It's not novel. It's actually just a reiteration of what its normal function is during development. So in tumors, you have undifferentiated tumor cells with the neural crest gene signature, and these cells can proliferate. They're not differentiated. If you get rid of BMP signaling, MITF goes up, and the tumor cells start to differentiate. In development, you have neural crest cells that a proportion of them are being, when you lose GDF6 driven BMP signaling, here MITF goes up and you get an excess of differentiated melanocytes. So it's essentially the same function. The tumors are essentially co-opting that function that is normally used by BMP signaling during neural crest development. All right. And one of the reasons why this is interesting is because there's been some fairly recent work that showed that one of the hallmarks of melanomas that are resistant to immune checkpoint blockade is that they're un- or de-differentiated. And so this has been shown in a variety of different ways. So both tumors that are resistant to immune checkpoint blockade, but it's also been shown in tumors that are resistant to RAS MAP kinase signaling blockade as well. So a de-differentiated phenotype is important for drug resistance. And what we were interested in finding out is if we target our pathway, if we target our gene, will these cells differentiate, and is it a viable therapy? All right, so there are a few things that we wanted to find out before we started targeting this, and here we're talking about targeting BMP signaling and potentially the GDF6 protein itself. So we looked to see what the BMP activity looks like in tumor cells when we take down levels of GDF6 and pathway activity. So these are cells right here. These are A375 melanoma cells. This is a fairly weak shRNA. This is a stronger one. You can see that with the weak one, the phospho-SMAD levels go down. With the stronger one, it's mostly gone. And you could see also this effect if you do ChIP-seq, so that in GDF6 knockdown cells, you lose a lot of the binding of phosphorylated SMAD on its targets. So that says that basically GDF6, although it's one of many ligands, it's regulating most of the BMP activity in these melanomas. What about the expression of GDF6? So this is work that we did ourselves where we showed so this is GDF6 protein staining right here. It's cytoplasmic. This is a melanoma here, and these are normal melanocytes. You can see that it's up highly in melanomas as compared to normal melanocytes, and about 80% or so of melanomas have high levels of GDF6. If you go into Protein Atlas and look at GDF6, you can see there what they see is a high proportion of tumors have high levels of GDF6 as well. Now, what about BMP activity? So phosphorylated SMAD. This is phosphorylated SMAD right here in a tumor versus normal melanocytes. And in this case, this is a nuclear stain. And you can see here that phospho-SMAD is present in these cells, whereas it's not present in normal melanocytes. And again, it's about 70 to 80% of melanomas have active BMP signaling in them. In addition, you could see that the signaling is active very early on in tumorigenesis. This is a primary tumor, and you can see clearly that these tumors are expressing it as well, and it's about 75-ish% of those tumors. So it's on in a large fraction of tumors, and it comes on early. And then the last thing is, what does this expression correlate in some way to patient outcome? And to ask this question, what we did was to do a tissue microarray study where we stained for GDF6 and asked whether the levels of GDF6 correlate with patient survival. And what you can see here is that patients whose tumors have low levels of GDF6, they tend to do better than patients whose tumors have high levels of GDF6. So there is a correlation between high levels of GDF6 and poor survival for these patients. All right, so then we decided, okay, let's try to target this thing. And I mentioned before, there's small molecule BMP inhibitors. We can skip ahead, there we go. So there's small molecule BMP inhibitors, and the way that they work is by inhibiting the receptors, so the type 2 receptors. And so one option is to say, okay, well, let's use those drugs, or let's develop similar drugs. But there's a couple of problems with this approach. So one of them is that the existing drugs have very poor pharmacokinetics and pharmacodynamics. Basically, they're not present in the system for very long. The other thing is that the BMP signaling is on in a lot of normal tissues. And so in this case, what you're talking about is a pan-BMP inhibitor, and you run the risk of toxicity using those types of drugs. The other option that we considered was to do a monoclonal antibody that would specifically bind and neutralize GDF6. And so that's the approach that we've taken. And this has a variety of benefits. So one of the benefits is that it would be a cytotoxic therapy. I told you that when you knock down GDF6 or BMP activity, the cells start to differentiate, they ultimately die. It would potentially be broadly effective in melanoma, so the target is expressed in about 80% of melanomas. It would have low on-target toxicity. So GDF6 is a developmental gene. It's not expressed in adult tissues, except there is some expression in placenta, and there's some expression when there's bone regrowth. But that's essentially where it's expressed in adults. And those situations, of course, you can avoid treatment under those conditions. And so if you put in a drug that targets GDF6, it could have a very specific tumor effect, because it's really only the tumors that are going to be expressing it. And like I mentioned, it has the potential to target ICV-resistant tumors as well, because they're dedifferentiated. Okay, so to target this, we wanted to know, we're talking about an antibody here, and what needs to be the case there is that the target needs to be present outside of the cell. And so we wanted to confirm that. And in this experiment, what we did was to tag GDF6 with an HA tag, and then look to see if we could detect that HA tag on the surface of cells using FACS. So this is just control right here, so no HA tag. These are cells that were treated with media that had HA tag GDF6 in it, and we can detect that HA tag on the surface of cells. These are cells that are producing GDF6 that has the HA tag, and we can detect the HA tag on the surface of cells. So it's accessible on the surface. So it says that it's secreted. Some ligands don't actually make it out to the surface. We wanted to confirm here that this one does. Not only does it make it out on the surface, but it can act as an extracellular ligand. And to show this, what we did was some conditioned media experiments. So these are controls here. But basically what we did was to take cells that were treated with with an RNA inhibiting shRNA inhibiting GDF6, so we knock that down. And when you knock it down, you lose viability. And here what we did was to treat those cells with media where GDF6 was depleted. And you can see here that there's a reduction in viability. It doesn't really do much to these cells. This media does not. But in knockdown cells that are treated with media that has normal levels of GDF6, the viability is better. In knockdown cells that are treated with media where GDF6 is enriched, they do even better. So that's saying that the GDF6 that's provided by the media is acting to promote the viability of these cells. So it's acting extracellularly. So we went ahead and made antibodies. The GDF6 protein is produced as a proprotein, so you have this long N-terminal piece here. It's cleaved by furin proteases, and then this is the business part. This is the ligand over here. We made our antibodies against that ligand. And so what you'd expect is that if you look intracellularly, you should see this large protein here, and in addition, you should see the ligand form. If you look in extracellular media, you should really only see this. And that's essentially what we see right here. This is the proprotein right here, using our antibodies that we made. Here's the ligand form, whatever's present inside of cells. And then in the supernate, you can see that the ligand is there as well. And here we're looking at antibody specificity. This is cells that have been stained with our antibody. These are control cells, and then these are cells where GDF6 has been inhibited. You can see that the staining is way down, so that again says that there's some specificity to these antibodies. We stain tumors with our antibodies, and pretty clearly this is just a control antibody. This is the tumor right here. You can see that it's staining very high levels of GDF6 in this tumor. So we can see tumor-resident GDF6 with our antibodies. Now, what do the antibodies do in terms of BMP signaling? So in a series of experiments that I'll show, we're comparing the effect of our antibodies to the effect of these small molecule BMP inhibitors. And in this case, we're looking at phosphorylated SMAD. This is control cells, so pretty good levels of phosphosmad in A375 control cells. And here, these are cells that have been treated with the anti-GDF6 monoclonal antibody. And literally all that we do is we just put the antibody into the culture media, let it sit for a little while, and then do these measurements. So it's very much reduced, similar to what you see when you treat with the small molecule BMP inhibitor. We did experiments where we looked at the effect of the antibody treatment on a BMP reporter. This is the reporter right here. It's got these phosphosmad binding sites here, and it's driving mCherry expression. When we treat cells with our antibody, the expression of the reporter goes down. And in addition, it's similar to what you see with the small molecule inhibitor. So it's inhibiting BMP signaling, it's inhibiting its transcriptional function. And then we also were interested in looking at those differentiation genes. What's the effect on the differentiation genes? And here, MITF we're looking at. When we treat with our antibody, remember GDF6 and BMP signaling is normally inhibiting MITF expression. So when we get rid of BMP activity, MITF levels go up. And that's what we see here with our antibody, similar to what you get with the BMP inhibitor. We also looked at SOX9. This is the protein that's responsible for killing cells when you get rid of BMP activity. And again, we treat with our antibodies, SOX9 levels go up, similar to what you see with the inhibitor. Now, what effect does it have on cell viability? We're doing viability assays here, and so we're looking at different cells, different melanoma cells here. And sort of the workhorse cells that we've been using are A375 cells. I think that's pretty much most everything that I showed you up to this point. We also looked at SK-MEL cells, and so all of these cells have BRAF mutations. You can see that the viability of A375 and SK-MEL goes down when you treat with anti-GDF6 antibody. These cells are different in that they have very low to no GDF6 expression, so they're essentially control cells for us. When we hit them with shRNAs, they're pretty impervious to those shRNAs. So we expect that they would be impervious to the antibody treatment, and pretty much there's not much of an effect when we treat these cells, M14 and C32, with our antibody. Again, similar results with the small molecule. We also look to see whether the antibody is working through the SMAD BMP pathway. These are viability measurements again. We treat with the antibody, the viability goes down, but when we restore BMP pathway activity, the viability goes up. Essentially another genetic epistasis experiment showing that this is acting through the BMP pathway. This is looking at essentially cell proliferation over time. So you can see that there's a pretty big difference as you take the experiment out over a few days. The ones that I was showing you before are really a 24-hour time point, but we're going out to 4 days here. So these are control A375 cells, and the ones that are treated with the antibody, they essentially they don't proliferate. So again, BMP inhibitor over here. And the reason why the cultures aren't proliferating is because there's a lot of cell death that's happening. Again, this is what we would have predicted. So here, control antibodies, anti-GDF6 monoclonals. We're looking at cleaved caspase 3 here. In cultures that are treated with GDF6 monoclonal, the level of cell death is significantly increased. So it's causing both the differentiation and the death that we have seen previously when we inhibit GDF6 activity, BMP activity. And these are mouse xenografts where we're treating tumors, we're treating animals with our monoclonal. And the way that we set up this experiment was to implant cells and let the tumors grow up to 100 millimeters cubed, and then we began treatment with our antibody. And you're looking at the change in tumor volume after that point. So control monoclonal antibody, those tumors take off pretty quickly, but with the GDF6 antibody, you could see that they grow a little bit and then they start to tail off there. And so it has a pretty substantial effect on the growth of tumors in vivo. And so this got us excited. Oh, I'm sorry. So just to tidy this up, we looked at those tumors and we could see that the tumors that are treated with GDF6 monoclonal antibody, they have higher levels of cleaved caspase 3. So again, in this context, it's causing cell death. And the other thing that we looked at was the other question that we had was, is the effect on target? So what we would predict is if we treat with the antibody, anti-GDF6, that would take down BMP signaling activity, and we should see that using our readout of phosphorylated SMAD. And so that's what you're looking at here, phospho-SMAD 1, 5, and 8. In control-treated tumors, you could see that the tumor cells have pretty decent levels of phospho-SMAD. Again, it's a nuclear stain. But in the tumors that are treated with anti-GDF6, you could see that they have very much lower levels of phospho-SMAD. So this says that the effect of the antibody treatment is on target. It is hitting BMP pathway activity as we would predict. And so what we've done more recently is to take this a bit further, and we started out with monoclonal mouse antibodies, and we've humanized those antibodies. And the way that we did it, we worked with the CRO to do this, and what we did was to take the CDR regions and graft them onto a human backbone. We modeled the 3D structure of these antibodies, and in key positions, we did some affinity maturation, surface reshaping, that sort of thing. And we created 3 heavy chain variants and 3 light chain variants based on our mouse monoclonal. And we made all pairwise combinations of those. So we had 9 humanized antibodies, and we had a parental antibody that was pretty much the same CDRs that we had from the mouse. And so we've been testing those, and we looked at the affinities of the humanized anti-GDF6 antibodies. And here is our parental right here. And in this case, what we're doing is a quantitative ELISA assay. So here you can see the parental. There are some of the antibodies that are actually binding more strongly to GDF6 than the parental. and the others are a bit weaker binding. But in these cases, what we're talking about is binding at nanomolar or subnanomolar affinity. So that's pretty good, pretty good. We've done some viability tests, and so if we look at BMP inhibitor in this case, what we have is a reduction in viability. These are some of the antibodies that we tested out, the humanized ones, and you can see that they're also reducing the viability, as does the mouse antibody. And sort of the latest is that we've been doing these mouse xenograft experiments. They're essentially set up the same way, where we let the tumors grow up for a bit, then we inject the animals with antibody. And in this case, the ones that I'm showing you, this is the parental again, right here. We have one antibody that isn't working quite as well, but another one that's working maybe a bit better than the parental antibody. And so these are the antibodies that we're going forward with to do PK/PD tox studies, and hopefully we'll get them into the clinic. So to wrap up, I just wanted to give a brief summary and sort of implications here. Everybody knows about immune checkpoint blockade therapies. They've been a huge success in melanoma, but despite that, there is still a pretty significant unmet need. Last year, 2025, about 8,500 Americans died from melanoma, and worldwide that number is 60,000, at least 60,000. And so there is this unmet need. The anti-GDF6 antibody, it targets GDF6-driven BMP activity, resulting in tumor cell death, tumor cell differentiation and death, and we showed that these antibodies, they blunt tumor growth. Again, GDF6 inhibition, it causes differentiation. Is this a way that we could potentially treat the immune checkpoint resistant tumors? So these are the questions that we're asking right now in the lab. All right, so I'll just finish up there. So these are some of the key people in my lab that have worked on this project. So some of the historical stuff that I talked about, the initial identification and characterization of GDF6 was done by a really great grad student named Arvind. Alec was an MD PhD student in the lab, and he developed the mouse monoclonal antibody that I showed you. And then Oluchi is currently in the lab. She's a technician, and she's done the work on the humanized antibodies. And we've had really great collaborators along the way, mostly UMass folks. And our translational work, basically the humanization was supported by Bridge Fund. So this is the translational office at UMass. And we've also been supported through other mechanisms as well. And that's it. So thanks.
Fantastic, great. Questions?
Great work. This is very exciting. So I was wondering, with the antibody, the mouse antibody, have you tried in the syngeneic model to see if you can actually enhance the checkpoint inhibitor?
Yeah, so we'd love to do that experiment. But the syngeneic models like B16F10, Yum 1.7, those models, they don't have BMP activity in them. So they're in that class that is low BMP. So they're not an appropriate model for us to do that in, unfortunately.
Yeah, because before you can really go into clinical trial, they actually really want to see some in vivo work. Yeah.
So we've done those mouse studies where we put the antibody in and we show the tumor shrink. We just aren't able to do them in combination with, But what we're doing now is there are some in vitro studies that you can do to look for cooperativity. So we're gearing up to do those.
Very interesting. I have a general question. How often ligands are targeted by antibodies? Is it how long in general?
Yeah, I mean, there's a fair number of drugs that are out there that target ligands, like Humira, right, is a TNF inhibitor. Right? That's a ligand. There are actually 3 or 4 TNF inhibitory drugs out there, and there are other ligands that are targeted as well. So it's not as though we've come up with the strategy ourselves. It's sort of an approach that has been used, and there's some precedent out there. Yeah.
How specific is your ligand to melanocytes? It looks like in your fish that it's very melanocytic specific. You don't seem to have an effect on the two other pigment cells in zebrafish.
Yeah.
And whether or not this is something that can be used the other way, where you have melanocyte deficiencies, such as in vitiligo.
Yeah.
Is there any application there?
So, how specific is it? So, in fish, it's expressed by the neural crest, and so it's signaling. It's expressed by the neural tube, and so it's signaling to the neural crest cells. So a lot of neural crest cells are seeing it. And what I didn't show, we did some work to show, okay, well, what's happening in the case of when you inhibit it, right? So we see melanocytes go up. Well, what we see is that iridophores go down, right? The quantity of iridophores goes down. So essentially, what it's regulating is this cell fate decision between a melanocyte and an iridophore. So when you get too many melanocytes, you get too few iridophores. So that's kind of what's happening during development. Whether it would normally act on these other pigment cell types, I don't think we've done anything that says that it's specifically acting on those cells. I don't think we have. But you asked about vitiligo as well. We haven't really done anything with it. I have to say that we're very interested in vitiligo because I didn't talk about it, but I've been talking with other people here. We've done work where we've isolated human melanocyte stem cells, and we do this from panulectomies, so excess skin that's taken off from surgeries, and we identified melanocyte stem cells, and what we found, it's pretty new, but it looks like those stem cells have BMP activity in them. And so it's possible that inhibition of that BMP activity will cause those cells to differentiate. And maybe it could be something that could be useful in vitiligo. But that's like such hand waving at this point that, yeah.
Thomas?
Yeah, I'm not sure if I understood correctly, but so you also mentioned that this BMP can affect the bone regeneration.
Yeah.
Right?
Yeah.
Do you guys see any effects on bone health in general in the xenografts?
No, so GDF6 isn't expressed in normal bone growth, in adult bone. It's not expressed there. It's definitely needed during bone development, and so there are syndromic patients out there that have GDF6 mutations, and what they have are joint fusions, like in their extremities, fingers, toes, and so it's definitely needed for bone growth and bone development in developing embryos, developing people. But in adults, like in adult mice, when we treat them, it's not needed. It does come on, if there's a bone broken, it will come on, but that wasn't part of our study.
So I actually remember hearing years ago, maybe you can tell me if this data is real and has been borne out, that patients who were treated with BMP, like supplements to promote bone healing, had an increased incidence of tumors.
I don't know.
No, okay.
I don't know. I'll look at it. It sounds interesting. I'm not aware of that.
Okay.
Yeah.
Also, last question with the GDF6 antibody, does it also affect pregnancy, or if someone's pregnant, would you ever have some sort
No,
of?
you wouldn't do
No?
that. No, I mean most studies, at least in clinical trials, right, you have an exclusion for pregnant. But no, we wouldn't do that. There's no specific reason to, right?
Would it affect fertility?
We've never really done anything that says like, okay, you inhibit BMP signaling and the fertility goes down. We haven't done those experiments. I actually don't know the answer to that question.
there any other questions? If not, oh, one more? Yeah.
Yeah, so do you envision the patient population or something like this as people who are resistant to like nivolumab or any other inhibitors or anything like that?
Yeah, so at the outset, there's nothing, you would never get a trial, you would never have a trial where you would use this instead of checkpoint inhibitors, except in patients who are resistant. Yeah, because those are frontline therapies for stage 3, stage 4 patients. So yeah, you could look at patients that are resistant, and that's going to be that would be the initial phase 1, and maybe beyond. But yeah, and then you could ask questions. All right, is there some synergy? Can you use them together? That's those sorts of questions. But initially, we'd be looking at patients that are resistant.
Okay, if there's no other further questions, let's thank Craig for a great session. For the students, trainees having lunch with Craig, please head on up to the GSI conference room. Mike? Good. Thanks for coming.
Yeah, of course.
Oh my God.